Excellent. Okay. Well, thank you all again. We've now completed our very big section 1 of day 2 of the workshop, and I'm very excited to introduce the second section with one of our newest postdocs that was hired this year for the IEDB, which is really exciting. So I'd like to introduce Dr. Eve Richardson to the virtual stage to present B-cell epitope prediction. Thanks very much, Eve.
Great, thank you, Nina. I'll just share my screen. Can you confirm that that's sharing okay?
Yep, looks good. Thank you.
Okay, fantastic. So I'm going to be speaking today about our B-cell epitope prediction tools that are currently hosted on tools.iedb.org. And so just a brief overview of what we'll talk about. So obviously we've had quite a deep dive onto the T-cell side of things. So we'll just quickly have a very brief recap about what we're talking about with B-cell epitopes. And then I'll be talking about the prediction tools that we're currently hosting and are made available on the IEDB. And so the purpose of this presentation of what I'll be showing you today is trying to familiarize you with the interface that's available on the web server and what the outputs of that mean, and quite a high level of understanding of what each of the algorithms that we provide do. So we will not be talking about the standalone tools, which are available for the majority of things I'm presenting here today, nor the API, which is available for a subset of the tools. And if you're interested in that, please let us know for next year as well, obviously. And then we'll be talking about the tools sort of split up into two sections, the linear sequence-based epitope prediction tools, and then the 3D structure-based epitope prediction methods. And I know we've already had a couple of B-cell questions, so I'll try and sort of address those, the questions we've already had as we go along. So B-cell epitopes, I am sure you're all familiar with them and what we mean when we talk about B-cell receptors or antibodies. So the B-cell receptor is the membrane-bound form of the antibody, but people will often talk about them interchangeably. And the part of the antibody or the B-cell receptor that engages with the antigen is these variable domains, the VH and the VL, and those engage with the antigen here via the particularly hypervariable set of those variable domains, which are known as the CDRs. And then the part of the antigen that's engaged by the antibody is referred to as the epitope. You might have also heard of the paratope, which people often talk about with antibodies. That's on the other side. But for the purpose of the presentation today, we're kind of but excluding antibodies. So the prediction tools that we're presenting are not antibody specific. They will allow you to generally predict for a protein antigen, the most immunogenic regions. So there's one epitope for one antibody, but there can be many epitopes for a single antigen. And so with regards to features such as the host organism, as one of the questions was about chicken B cells, the host organism does not come into play with these prediction methods. So here's an example of what the data that looks like for lots of these models are trained on, which is very often these crystal structures where you can see the antigen and you can see the antibody, and you can see the way in which the antibody is engaging that antigen. And so the epitope we can highlight here in red. Yes, so that's the subset of the antigen surface that is interacting with an antibody. You'll also notice from looking at that the epitope residues in this instance are not continuous in sequence space, and that's what we refer to as a discontinuous epitope. There are also linear epitopes or sequential epitopes, and so usually protein antigens will contain a mixture where you will get subsections of the epitope that are contiguous in sequence, but very often you'll get these like disjointed epitope residues, which are variously known as sequential or non-sequential, or conformational. And indeed, the majority of B-cell epitopes are estimated to be discontinuous. And sort of whether an epitope is considered to be linear or discontinuous is very often a matter of the resolution of the method by which you've determined the epitope. So obviously, if you're doing stuff like microarrays, you'll be more likely to see epitopes in a linear context. But yeah, these are the two major categorizations of epitopes that lots of the epitope prediction tools that we'll talk about make reference to predictions of one or the other. So when should you use epitope prediction methods? So as I think Bjorn also has said yesterday, your first recourse should always be to experimental information. That will always be more specific to your antigen of interest than the predictions, which in this case will be trained on tens or hundreds of different antigens. So it's definitely in your interest to verify thoroughly that there's no information available in the IEDB. But even if you find some information in the IEDB, you may also want to sort of know all of the candidate antigenic determinants in your antigen of interest, other than the ones that other people have described experimentally. So there are sort of multiple scenarios in which you might want to use epitope prediction methods to understand your antigen better and understand how it's likely to be recognized by B cells. So now let's get into the tools. So you'll find this header B cell tools on the IEDB analysis resource page. And under that, you'll find the B cell epitope prediction tools. And today we'll be talking about these three headers. So the first one, prediction of linear epitopes, and then we'll have the two structural methods, DiscoTope and Ellipro. And we'll be doing a bit more of a deep dive into each of these tools. So as I said, we have the sequence-based epitope prediction, which was our first header, but we do also have the structure-based epitope prediction. And you sort of may be questioning when to use one or the other. So in previous years, often sequence-based epitope prediction is your only option if you don't have a 3D solved structure. And before, basically before, I guess, AlphaFold 2 and before structural modeling of antigens became much easier and much better. And because of AlphaFold 2, now very often you will be able to find a 3D solved structure or a structural model of your antigen. And because B cell epitopes must be on the surface in order to be recognized by a B cell receptor or antibody, the surface accessibility is a really critical feature, which you won't know from the sequence. So if you're doing sequence-based epitope prediction, you sort of have an extra prediction step that your model must learn, which is predicting which of the sequences are surface accessible. Whereas if you have a structure or a good model, then you will know with a higher degree of certainty which of your residues are surface accessible, as well as you'll know what the secondary structures, etc., are that we know to be associated with antigenicity in B cells. So the first set of methods are amino acid scale methods for sequence-based epitope prediction. So that we have a number of scales available, which are quite complementary, and they cover quite a lot of features that we know to be correlated with antigenicity in antibody epitopes. For example, the presence of beta turns, so hydrophilicity, which is a really big one. Flexibility, so there are two scales here that we make available in our prediction suite, and then surface accessibility as well. As I've said, this is a really critical one for B cell epitopes. And then finally, we have an antigenic propensity scale, which is calibrated on experimental data, what the occurrence of residues that we have observed in experimental data are within the epitopes. So that sort of allows you to, moving along the sequence over windows, combine these amino acid scales in various ways, most commonly just simply averaging over the window. And then, yeah, that is quite a strong form of prediction. And even as we get more complex methods using protein language models, for example, I think these are still kind of a good bread and butter of epitope prediction. These are things that time and time again we know are associated with immunogenicity in B cell epitopes. And so it's always good to look at your protein of interest in the context of these amino acid scales. And then moving on, we have the machine learning methods. And I know that we have already had questions by some participants who are clearly, up with the literature and know about some of the new methods that are coming out. So the machine learning methods, these are trained essentially. So rather than just being general feature scales, they're calibrated to particular data sets. So you have a positive data set and a negative data set that you train the model on. And then you can featureize that in some way. Most often, it has been traditionally amino acid scales. Now more people are using stuff like the protein language models. And then with those featureizations, you apply some machine learning models, such as a random forest for BepiPred 2.0, but also various other things, such as artificial neural networks. Many methods favor the support vector machine. And then through that, the model learns to distinguish the epitope and the non-epitope regions of the sequence, outputting a score for each region for you. And so while we're on the topic of this, and while we're talking about BepiPred 2.0, I should just clarify on questions yesterday that, yeah, indeed, we do have plans to incorporate BepiPred 3.0 onto our web server, hopefully before the end of the year, and will certainly be included in the next gen tools release for B cells when that happens. Okay, so now let's go back as to and have a look at how you would be doing this. So you click through into your link here, and your input, you can either put a Swiss Prot ID, or you can enter your sequence in plain text format. And then you can simply click which of these methods you'd like to do. So BepiPred 2.0 is a default and heavily recommended method. But you can also have a look and click through on each of these onto the help tab and read through. So we have the tutorial section under the help tab. And then if you want to look at the primary literature more, you can click onto the reference tab and have a read of the source publications for each of these amino acid scales and for BepiPred. So now say that you've read those and you're happy with BepiPred 2.0 as your prediction method. Let's see what the output looks like. So our example sequence here, we have the RBD region from the SARS-CoV-2 spike glycoprotein. We're submitting it BepiPred 2.0. We simply click the submit button, and after some thinking, it will return an output of this format, which I think you will be familiar with from Bjorn's presentation yesterday. So this graph, we'll just talk through that. So you have on the x-axis the position. So this is just the position from the first to the last amino acid that you input. And then on the y-axis, we have the score. So this is the BepiPred 2.0 score in this instance. And then the coloring is by threshold. So the default threshold is 0.5. And so all of the residues that have a score above that are colored yellow, and all below are colored green. And you can see what the minimum and maximum scores are. And so again, that threshold is adjustable. So if you'd like to highlight on the regions which are most strongly predicted by the model, you can increase that threshold so that it only highlights the most extreme predictions of the model. And so then you will also see that there's this table called predicted peptides, and these correspond to the linear epitopes, as we spoke about. So these are contiguous sequence segments where the score lies above the threshold that you have selected. So you kind of just walk along the sequence and take subsets of the sequence where the score is above the threshold. In this case, it has resulted in, I think, 9 predicted peptides, or was that 10? Oh yeah, 10. And you can look at those and you see sort of the maximum length, and do with that information what you will. Then you can also look at the predicted residue scores for each residue individually, and you can sort that either by position, or you can sort it by the score, and then you can see the assignment for epitope or non-epitope according to the threshold that you selected. But if you'd like to analyze that in in another fashion, you can also download this result, and that will download that for you as a CSV file, so you can look at that in Excel or your programming language of choice. And it often can be informative to look at multiple methods multiple prediction methods. So the ones that the IEDB has are generally they're complementary. So the beta turn prediction results, for example, will tell you something more about your antigen, and that will tell you something different from the flexibility prediction results, for example. So I think it's often, even if you only go on to use the model that you're most confident in, it still can be really interesting to look at these other amino acid scale methods and see how that correlates with things. So, for example, you can see in all of these methods, there's this sequence that seems to be a beta turn and has a high flexibility prediction and is also predicted by Bepipred 2.0 to be in the epitope. And so now we're going to move on to the 3D structure-based epitope prediction methods. So, as I said, you start off with an antigen structure or a model. And the advantage of that is that you know what the neighborhood is, like the spatial neighborhood of each of the residues. So instead of having to predict it, you can directly look at the structure and calculate that from the distances of the atoms. And we host two classes of structure-based epitope prediction methods. We have DiscoTope and we have Ellipro. And they work in, again, different and complementary ways. So DiscoTope for each residue looks at the neighborhood to calculate surface accessibility, and then uses these amino acid log odds scores, as well as the Parker hydrophilicity scale, to output a score for each of the individual residues using the structure that you have input. And I'll talk a little bit more about that. But then Ellipro is more focused on protrusion, so the residue protrusion index and how much kind of the residue is sticking out, essentially. So we can find that again under this tab of B-cell tools. You can click through onto DiscoTope. So an overview of DiscoTope. So originally this method was published in 2006, and then in 2012 there was an update. And both methods are trained on these 75 crystal structures of the antibody antigen protein complexes. And DiscoTope 2 was an advance in multiple respects, including the fact that it takes into account the fact that an antigen can have multiple epitopes. And then, as was alluded to in that figure, it assigns each residue a score, which is some linear combination of hydrophilicity, the amino acid propensity expressed in the terms of a log odds ratio, and then some measure of surface accessibility, either in terms of contacts or the upper half spheres measure for DiscoTope 2. So let's have a look at what it looks like. So DiscoTope 2, structure-based antibody prediction. The structure of the antigen, just to clarify. So you identify the structure, so you can input a PDB ID, and then the method works on a single chain at once. So you'll have to tell the web server which chain you would like it to perform the calculation on, and then you can select the version. But you, I guess, might be asking, how do I know if I have a protein structure? How can I find it? How can I find the chain ID? So the PDB is the number one resource for this. So previously, I think before last year, there was these just experimentally solved structures, and now there's this fantastic collaboration with AlphaFold DB, so that you can find 5 times as many computed structure models where people haven't solved the structure of your antigen of interest yet. And the way we would do it is via sequence similarity search. And you can, if you don't want to include the computed structure models, you can exclude them, or you can include them with this toggle. And you can actually just directly paste your sequence into the search bar, but we'll just show you what it looks like when you click onto the sequence similarity search. So what you'll want to do is you want to paste your sequence into the sequence similarity box. There's a return field as well, and it will prompt you to select polymer entities for that. And then you can toggle or untoggle whether you want to include the computed structure models, and then you can search. And this is examples of the 2 types of structure file that you can download from the PDB. So the top one, as you can see, the icon is a beaker. So that's an experimentally solved structure, something solved by either X-ray diffraction, increasingly cryo-EM, NMR, for example. And so that would be your preference, because this is experimentally solved. But there are many proteins for which there is not a solved structure. And in that case, so for example, this salmonella protein, you can find the AlphaFold model through that. So then just to show you how to download these files, you can click through onto this entry 1LVH, and then you can just simply click the PDB format under download files, and then that will download the file for you. And you can input that directly into the IEDB. But as we said, you need to identify the chain. Again, you can just scroll through onto the through the PDB entry, and you can look through there under the macromolecules tab, and you can see here that there are 2 chains that correspond to our protein of interest. So you can select one of those, A or B. It's maybe slightly less intuitive for the for AlphaFold DB, because if you click the same button to try and download, you'll get this file format that that you might not necessarily recognize, although this MMSIF file format is becoming is now the standard. But in any case, you have this model CIF file. The IEDB cannot accept this currently, so you cannot download this directly from the PDB. But you can still use the PDB to verify which chain you need to input into the IEDB interface. So this is A for monomeric proteins, and indeed if you model these things yourself with AlphaFold, these things are modeled alphabetically. That's just a little extra detail. So you can get the PDB file if you click through onto AlphaFold DB. So if you click through onto that, then you'll get some extra information from AlphaFold DB, including the PDB file, and that you can directly input into our web server. So hopefully that's explained how you can find structures using the PDB and using the AlphaFold DB. So that hopefully will enable you to use the structure-based antibody prediction tools that we have available. So now we're going back to DiscoTope. So now we have our PDB ID. We want to select the version. So we would recommend selecting the most recent version, DiscoTope 2.0. And by default, there are threshold values selected, and these actually produce comparable sensitivity and specificity values. We'll click submit, and then we have the same this consistent output format where you have this threshold that you can change. You can again save the prediction as a CSV, and you have the position on the x-axis and the score on the y-axis. And if you click through into the table view, you can get this a similar tabular output sorted by default by residue ID, which will tell you your DiscoTope score, as well as some other things that you might find interesting, such as the contact number, which is something that's used by DiscoTope 1, for example, or your propensity score, which is calculated based on the amino acids. And yeah, so this is a tabular view that when you click save prediction, that will download as that. And yeah, the positive predictions are displayed in green. So those are all the predictions that all the residues that have a score above 3.7. There's also a great thing with the 3D view. So you can have a look in the 3D viewer where the epitope is. So you'll see that it's on the surface. And it's highlighted in yellow. The different colors are the different chain IDs. So you can have a look at that through the viewer, which is a nice detail. Okay, so now I'm going to talk about Ellipro, which is the epitope prediction tool based upon structural protrusion. And so this is a method that dates back to 1986, even the theory behind it, but the actual algorithm itself was developed in 2008, and it still continues to perform really quite well in benchmarks. So Ellipro predicts both linear and discontinuous antibody epitopes. And as part of the algorithm, there are 3 sub-algorithms. So the first thing it does is you can approximate the protein shape as an ellipsoid, which is part of Janet Thornton's method. And based on the ellipsoid, then you can calculate for each residue a protrusion index. And then the protrusion index, you apply a cutoff on that. So if you have a cutoff of 0.5, everything with the protrusion index above 0.5 is then a candidate epitope residue. And then there's a clustering of the neighboring residues based on some distance between those residues in order to produce a set of conformational and linear epitopes that make sense given distances and given the protrusion indices. So this is the interface for that, very similar. So you can identify your structure or again upload your file. So that can be PDB ID or your PDB file from the PDB or from AlphaFold-DB. And then you can input the minimum score, which is by default 0.5. And the higher the score, the fewer epitope residues you'll predict. And then you can specify a maximum distance. And yeah, we have a default value of 6 angstroms here. And those are both things that can change the number and the distribution of epitopes. So we'll click submit, and then we will be prompted to select a PDB chain. So you can select both, or we can select a chain of interest, but it's yeah, it's worth noting that the calculations are based on each chain individually. And so you click submit, and you have a table for your predicted linear epitopes, as in the previous methods. And then we also have your predicted discontinuous epitopes, which are in the format as you may be familiar from the IEDB data. And so the predicted linear epitopes you can see there, and then the scores are the average scores for the residues presented in the epitope. So that's a score for the total epitope. And again, you can view these within the context of the 3D structure. Or, sorry, here, you can click to view the residue scores. So yes, you'll be probably familiar with this format now of the graph and of the data table. So I think, yeah, we're at the point of summarizing. The IEDB hosts numerous B-cell epitope prediction tools, which are, complementary to one another, and which, correlate with what we know about B-cell epitopes in general. Our currently recommended sequence-based epitope prediction tool is the machine learning model BepiPred 2.0. Though, as we said, BepiPred 3.0 has really made quite a good advance in performance. So when that becomes available, hopefully, towards the end of the year on the web server, we would recommend using that. More generally speaking, with the current tools available, if you have a good model or if you have a structure of your antigen available, we would certainly recommend at least looking at that through the structure-based methods to confirm that any prediction methods made by, say, BepiPred 3, when that becomes available, do actually correspond to surface accessible regions. So those are our methods, DiscoTope and Ellipro. And yeah, hopefully we've made it clear to you how you can use the PDB to retrieve those structures to input, as well as the computational models when structures haven't been solved yet. So that's everything from me. Thank you for your attention, and we'll be happy to answer your questions.